Welcome everybody. Let me just start by sharing the screen. Is the first slide visible?
Yes.
Excellent, excellent. Thank you so much. It is indeed a privilege to be able to present in this forum and to talk about, one of them, of course, is somebody I personally know very well. So, as part of my stint that Professor Viswanathan mentioned at the Lawrence Berkeley National Laboratory and the UC Berkeley, my postdoctoral research was in glycobiology of cancers and development. So I was looking at how sugars are involved in development and cancer. And while I was primarily supervised by a giant in cell biology, Professor Meena Bissell, the work happened in extremely close supervision and mentoring with Carolyn, who was one of the most amazing people I've come across academically and beyond that. So there are a lot of very personal aspects to what I'll be talking about today. Of course, relates to more to her work and the larger ideas of the click chemistry and bioorthogonal chemistry. So I start with the slide, of course, because the award this year was won by three brilliant individuals and sort of three legends, if you will, in and it's in fact also illuminating and humbling on a different level that the work that's been lauded is so wide ranging and so impactful that somebody who has a clinical training like me working in a genetics department in India gets an opportunity to talk about what is essentially a fundamental discovery in pure chemistry on some level. So that's the groundbreaking aspect of this work, as I'll try to show a little bit of in the next few minutes. So let me start with talking about I'll in most of this is, of course, I'll be working with material that has very eloquently been brought out by Carolyn, by Barry Sharpless, who's of course for whom this was his second Nobel, and the language that they use is so illuminating that at times I'll just simply quote them because there's no way to better paraphrase the thoughts that they have had in these concepts, and I won't be able to explain them any better than they can, and they're the words that they choose. So, let me start by saying that the context of this work comes back to, I guess, the late 90s, when combinatorial chemistry was at its helm, where one was reasonably able to synthesize a lot of molecules, a wide variety of molecules, and a lot of chemistry was about being able to develop libraries of compounds of different structures and orientations that would be able to be deployed in order to give rise to different functions. So structure function relationships were being explored to a great extent. But the challenging question was, we can synthesize a bunch of things, but can we synthesize them now under aspects that make it economical to do so? Because now, these kind of chemical synthesis was increasingly beginning to impact pharmacology, being able to impact medical biology in so many ways, and therefore, as sort of things move from academia into industry and sort of into the factory, into the foundry, other dimensions start coming into place. So the questions that were being thought about was how efficient are these processes, many of which were multi-step processes. Can we make things more efficient? Can we increase the speed by which we can synthesize more and more complex compounds? Can we do so in more cheaper ways? And can we also do so in a manner that, once we make these compounds, extracting them becomes tractable at the cost of producing waste products that need to be removed in some way, and we have to sort of sift off the what is valuable from what is waste. Also, the way and what is valuable has to be produced in amounts that are practical to be able to scale up. So, in terms of scalability, these were questions that were plaguing a lot of at the edge chemists at that time. And so, to ask such to sort of more philosophically handling this at that time in conferences, and then subsequently in this very influential, brilliant paper, which I recommend anybody interested in this topic to actually get started with reading, was this paper by again, a senior co-author being Barry Sharpless, and his students, Hartmut Kolb and Finn, which came in Angewandte Chemie in 2001. And I will simply do a mini recitation here by just reading out the beautiful abstract of this paper. And so let me do that. So, examination of nature's favorite molecules reveals a striking preference for making carbon heteroatom bonds over carbon-carbon bonds. Surely no surprise given that carbon dioxide is nature's starting material and that most reactions are performed in water. Unsurprisingly, given that most of the compounds, for example, in biology used in living matters, of course, are compounds which have carbon with nitrogen, oxygen, hydrogen, and so on. Nucleic acids, proteins, and polysaccharides, of course, being complex polymers of sugars, are condensation polymers of small subunits stitched together by carbon heteroatom bonds. They're talking about peptide bonds, glycosidic bonds, and so on. Even the 35 or so building blocks from which these crucial molecules are made, each contain at most 6 contiguous carbon-carbon bonds, except for the 3 aromatic amino acids. Taking our cue from nature's approach, we address here the development of a set of powerful, highly reliable, and selective reactions for the rapid synthesis of useful new compounds and combinatorial libraries through heteroatom links, CXC, an approach we call click chemistry. Click chemistry is at once defined, enabled, and constrained by a handful of nearly perfect spring-loaded reactions. The stringent criteria for a process to earn click chemistry status are described along with examples of the molecular frameworks that are easily made using this Spartan but powerful synthetic strategy. So lucid as well as so profound as an abstract. So moving ahead, as you can see, Sharpless seems to be again inspired by the living world in terms of this new approach. So this was at that time in 2001 when this paper appeared. It was more of a hypothesis, more of an idea shift that he was envisaging in the chemical world. And for this, they seem to have dipped into the complexity of living molecules. So as shown in the abstract, for instance, if you look at starch and the structure of starch as shown here, you would see that, of course, you get these sort of 6 carbon elements repeated one after another, with joined by these carbon, oxygen, carbon bonds, and so on. And this is a theme that gets repeated whether you talk about sugars, whether you talk about amino acids, which are, of course, brought together to form proteins, and then nucleic acids. And so this theme of having, as he mentioned, the carbon CXC bonds or carbon heteroatom bonds is some kind of a theme that gets repeated more and more as you look into the living world. And it gives rise to very complex forms as you move forward. So then the question comes, and this is something that, Sharpless, in a very, very ironical fashion, you can actually feel the irony in the way he writes, is that it is almost seductive for chemists to be inspired to try and mimic the complexities of what might go on in the process of bringing together these biological molecules, and hope that one can sort of play God by actually trying to be able to replicate the same process inside a test tube and try to see whether the same complex way in which these complex molecules are fraught in the bodies can actually be done so in the laboratory. And this is something that Sharpless seemed to revolt against in a very interesting way. So he said that we have to probably move away from trying to think we can imitate this. There are easier ways of getting to this complexity, far more interesting ways of getting to this complexity, rather than going through this fashion. Why so? Because Sharpless very interestingly boils this down to mostly what he calls, of course, with Finn and Kolb. I don't want to, this paper is a three-author paper, of course, in that sense. So what they talk about is that much of this chemistry is carbonyl chemistry. It's essentially chemistry where you have a carbon which is linked to oxygen. And that's where most of these linkages and heteroatom linkages are taking place. And carbonyl chemistry pretty much defines a lot of what happens in biology. For example, if you want to look at how glucose is synthesized in this very famous cycle called the Cori cycle, where glucose and lactate can be interchanged within our bodies, once in the muscle and one in the liver, where in the muscle you have basically glucose being converted from a 6-carbon sugar, it goes to a 3-carbon compound, and then in the liver, where it goes from a 3-carbon compound to a 6-carbon compound, you basically have synthesis going on in the liver, and in this process you have an enzyme coming in called the aldolase, which plays an important role in combining dihydroacetone phosphate and glyceraldehyde 3-phosphate to form fructose 1,6-bisphosphate, which eventually gets converted to glucose. So basically, you have a lot of these very interesting enzymatic chemical reactions that are involved. And so, of course, those of us who have looked at or studied biochemistry textbooks and have looked at all these very interesting pathways of anabolism, what we call in metabolic terminologies as anabolism, which is basically synthesis in a biological jargon, involves a series of enzymatic reactions which are continuously producing and building up these structures in a manner such that you get, there you go, so in a manner such that you start getting these big complex compounds that can be subsequently utilized for various processes. And our understanding and our breadth, if you will, of anabolic pathways and catabolic pathways, degradation and synthesis occurring within the living world, has gone on to humongous levels. So again, what they say is that with carbon dioxide as a carbon source and very few good carbonyl chemistry-based reaction themes, nature achieves astonishing structural and functional diversity. So that's their thesis, and that carbonyl chemistry is therefore used to make a modest collection of approximately 35 simple building blocks, which can then be assembled into biopolymers. So they talk about the various amino acids and the glycans, which can be brought together to form these big biopolymers, which are the mainstay of how we behave, how we are made up of, and so on. The enzymatic polymers serve in concert with increments of energy provided by adenosine triphosphate as selective catalysts, which prevent nature's carbonyl chemistry-based synthesis from collapsing into chaos. Since many biosynthetic pathways require a unique enzyme for each step, the enzyme control strategy requires a huge investment of time and resources for catalyst development. And here's again, the irony comes in. With a few billion years and a planet at her disposal, nature has had both time and resource to spare, but we, as chemists, on a human time scale, do not. Okay, so what they say is what they propose is that carbonyl chemistry and the brilliance of carbonyl chemistry to be able to give rise to all the complexity that is involved in living life it can be explained through evolution, through the long stretch of time that it has taken for evolution to be able to evolve all these compounds, put them in the right place, bring them together in the right compartments within the cell, and be able to efficiently chug out all these molecules in time. We cannot do that in a laboratory. So, and there are other aspects to these as we go along, and as we see, for example, in our lab, we worry about the process called diabetes, for example, where we know that excess glucose, excess fructose, and excess other compounds that are present in our body and cannot be with age or with other kinds of diseases cannot be removed, can play very bad roles in terms of all the pathologies that are associated with, say, diabetes. For example, the blindness, the kind of kidney disease we have, can be understood through this interesting molecule called methyl glyoxal, which actually is a waste product of glycolysis. Glycolysis, the same glycolysis which occurs in every cell of our body, by which you can actually very quickly generate pyruvic acid from glucose. Okay, so the point here being that even the processes that we read of in textbooks, classical processes that we think are extremely efficient processes, need not be very efficient either. Even despite the billion years of evolution, there are waste products of existing processes in our body which are also inefficient in terms of the fact they're not clean. A lot of dirty byproducts are formed that can create havoc and which have very deep and ominous consequences for us. So, which basically strengthens the point that can we think of alternate approaches by which we can synthesize and create new reactions. Moreover, the fundamental and lasting objective of synthesis is not the production of new compounds, but the production of properties. Again, another very interesting quote of George Hammond that Cole, Finn, and Sharpless invoke to tell us basically that, again, don't be contented and seduced by what is being produced in the biological world by the biological cells, because ultimately what you need is not new compounds but new properties that have not been even evolved. We would need those properties in order to get to new chemistry. And bioactive natural products that have most intrigued synthetic organic chemists have frameworks which are difficult to construct, largely because there are too many contiguous carbon-carbon bonds. However, those are not the only kind of molecules that can have useful biological effects. One can have new compounds with new properties and therefore new structures that can have consequences and can play important roles as as pharmaceuticals, as important diagnostic tools, as prognostic tools, and so on. So this is the backdrop in which click chemistry comes in, and then remember, this is just an idea platform at this stage. That this paper was all about ideas, and what they say is that all searches must be restricted to molecules that are easy to make. So I think it's the brilliance of a Nobel laureate that thinks simplicity to fight complexity. So click chemistry, they define, is an approach to joining small units together with heteroatom links to ultimately develop a set of powerful, selective, modular blocks. So these are very simple reactions, and then they have a series of criteria by which they define these, or they necessitate the inclusion of reactions which have been studied before as well, which can be brought into click chemistry. So what are those stringent criteria? They had a very stringent criteria list because, of course, this was in the hypothesis domain at that time. So these reactions should be modular. In other words, they should be simple, clean, and independent with respect to other chemicals around them. Wide in scope, high yield, inoffensive byproducts that can be removed by non-chromatographic methods. So all of those limitations that I talked about sort of come in terms of what click chemistry should be, and whereas which other types of methods could not. They should have simple reaction conditions in the sense that they should be insensitive to oxygen, water. The solvent, there should be no solvent, or at least a solvent that is benign as water and so on, that can be easily removed. And so basically product isolation should be simple, and purification also should be as simple as possible, such that, and the product must be stable under physiological conditions. So these are the sort of stringent criteria, the set of desirables that click chemistry should have. But most importantly, which Professor Viswanathan also referred to, is the fact that they should be driven by a very high thermodynamic driving force, in the sense that they should almost be like the way you put your seat belt in a car, that it clicks in a thermodynamically driven fashion. But if done so, click chemistry typically is irreversible. So it cannot be reversed back. Okay, such processes proceed rapidly to completion and tend to be highly selective. Those two processes, again, I think the metaphor of Lego is very apt. It's a highly efficient, thermodynamically driven, specific reaction leading to a very clean product, which is the combination of these two things which get sort of clicked together. And so we think of these reactions as being spring loaded for a single trajectory. Okay, so it's as if it clicks together only in one way, producing a clean end product. Okay, so these were some of the sort of ideas that came in. So there have been a bunch of reactions which have been described as being important for click chemistry, but the one reaction that has sort of stood the test of time, which has almost become synonymous with click chemistry and of course gotten popularized for various reasons, is this particular reaction called the Huisgen cycloaddition reaction. Rolf Huisgen was one of the influential German chemists in the previous century. And that's called the 1,3 dipolar cycloaddition reaction. So what this involves, in simple terms, this involves the coming together of an azide and an alkyne. The azide being the 1,3 dipole compound and the alkyne being sort of a dipolar of 5, which seeks the dipole and then quickly gets conjugated to it. So you can see how you have these 3 nitrogens here and the dipole here, and then what happens is that the 3 nitrogens get combined with the 2 carbons to form this 1,2,3 triazole. Okay, the pH of course being phenyl. So these alkynes can be either, or it could also be alkenes. Alkenes can also theoretically participate in these processes. So they need not necessarily be terminal. They can also have, if in case of alkenes, you can have another sort of conjugation here as well. But then eventually the structure that is formed is this very stable 1,2,3 triazole compound. So this is the one of the major sort of cycloaddition, 1,3 dipolar cycloaddition is one of the almost becomes synonymous with click chemistry, and is probably the poster child of click chemistry. Okay. Now, the interesting part, of course, is that this is not a new reaction. It has been shown in the past. It was there in the past as well, where such products with various, of course, side chains containing the alkynes have been used to make the cycloaddition reactions. And you can see the sort of the generic term is out here, where you have a terminal alkyne linked to this particular azide, and upon heat you get these 2 different sort of isomers of the 1,2,3 triazole. So while there is some specificity, you can see that there can be 2 products formed, and this requires heat. So here's the interesting part, and that's where Meldal and Sharpless's group came together. Although they did not come together, they independently were able to take it to the next level when in a couple of years after that paper of Sharpless's sort of the ideation paper came out, they were able to show that such reactions can be click based and can form, instead of forming 2 isomers, a single product, and also can take place at room temperature if you have copper as the catalyst, cuprous form. Okay, so it could be copper sulfate with ascorbic acid, which reduces the copper and make it cuprous. And in presence of, they showed it in presence of butanol. Meldal's group was able to show this as a solid peptide synthesis. So what they had was the alkyne attached to this phenylalanine glycine groups, and then you could basically see that you could get this cycloaddition reaction going on and forming a stable 1,2,3 triazole product. And same thing in case of Sharpless's group was able to show a similar process was going on as well, both at 25 degrees. So basically, this several criteria for click were met by this particular reaction, which they call the CuAAC, or the copper added alkyne azide cycloaddition. Okay. Now, what is the inherent problem with this. Great. This was groundbreaking. So the first click chemistry reaction could be performed in the laboratory, but if you could doing this in biological settings was a problem. And the reason is because of the inherent disadvantage of these kind of reaction milieu is that copper or cuprous forms are inherently toxic to living organisms, cells, and so on. But here is where again people dipped back into history and were able to find that there were that again brilliant chemists like Wittig and Krebs had shown that certain cyclic alkynes could act as very efficient dipolarophiles with azides. And the reason for the high reactivity is the strained nature of such dipolarophiles, which is released by cycloaddition, for which you do not need the copper. And that's what Caroline was able to show. Caroline's Bertozzi's group in UC Berkeley in the early part of the century was able to show that this strained cyclooctane, if this was presented to the azide, it acted as a very good dipolariphile, and you would, although you would still get a mixture of isomers, you would still nevertheless get the 1,2,3-triazole cycloaddition product. Okay, and this process was known as strain-promoted azide alkyne cycloaddition. This did not require copper. So this was another big jump that occurred. And as you can see, how each of these giants are building on each other's work to take it to the next level and to the next level and to the next level, moving with very fundamental chemistry, but getting now into biochemistry, and as you will see, getting into beautiful cell biology. But before we get there, let me talk about a little bit about how Bertozzi's group was able to sort of bring this into relevance, into specifics. And this requires us to understand the presence of, or the complexity of sugars that are present within our body as part of our cells and so on. So sugars typically come in two ways. We think of sugars in two ways. One is, of course, in terms of metabolism, one in terms of utilization of energy, glucose being the standard sort of currency which gets degraded and then you gets to form acetyl coenzyme A, which gets into Krebs cycle and is ultimately moved into the electron transport chain where you get ATP and so on. A second role of sugars is in the form of structural constituents of cells. They form the structures of the body. And the way that happens is that different sugars or glycans, as they call, are eventually form these long polymeric structures that are then conjugated to proteins and lipids and brought to the cell surface, sometimes even inside the cells, but mostly on the cell surface, where they form the outermost coats of our cells, and that is known as the glycocalyx. Important sugars in this process are, well, not so much glucose, but N-acetyl glucosamine, N-acetyl galactosamine, galactose, mannose, fucose, and a 9-carbon sugar called sialic acid. Sialic acids are very interesting because if you think of this sugar coat on the surface of cells, it looks like a bunch of trees with leaves coming out because there's branching as well. Unlike proteins, sugars can branch. Okay? So it forms these tree-like patterns, and the last leaf, the outermost leaf of these trees are these 9-carbon sugars called sialic acids. How is sialic acid formed? Very interestingly, sialic acid can also come from glucose. Glucose forms this process, this particular molecule called UDP-glucnat or N-acetyl glucosamine, which gets converted to N-acetyl mannose or N-acetyl manosamine. Okay. N-acetyl mannose subsequently, of course, gets converted to 6-phospho N-acetyl mannose, and then subsequently forms this particular molecule called N-acetyl uraminic acid, which is sialic acid, which ultimately goes into the nucleus, gets conjugated to a CMP, and then CMP sialic acid eventually gets into the Golgi body and comes out to the cell surface. And there you can see this pink colored thing is the sialic acid, which is sitting on the surface of the proteins, which have these sugars on their surface. Okay, so basically, mannose or N-acetyl mannose is very important to the formation of sialic acid. So what the Bertozzi group did was something very interesting. You can cheat this reaction and hijack this process by doping it with like a cheater. What is that cheater? So instead of manosamin, you can basically add per-acetylated azidomanosamin. Per-acetylation allows the molecule to get inside the cell. And once it's inside the cell, azidomanosamin now participates, just like mannose or manosamin, it participates in the sialic acid biosynthesis. So it just gets hijacked into the biosynthesis pathway. As a result, what happens is you basically get this azidosialic acid of sorts on the surface of the cell. So instead of sialic acid, this sialic acid now has an azide attached to it as it comes out. Not all of it will be there. There'll be some sialic acids which will have the azides along with. So you can see the protein is marked in purple, and then you have this branch sugars with the last sugar being sialic acid. Now this sugar is not a pure sialic acid, is azidosialic acid. Okay, so what they have done is they have brought an azide to the cell surface. What do we need now? Remember your click chemistry? The cycloaddition was azide alkyne. So now if you bring in an alkyne, here, the alkyne will come and click to the azide on the cell surface. And that's precisely what the Bertozzi group did. So these were early papers where, for example, these are cells. These are called Urquhart cells. They are a particular type of cells where what they did was they used this difluorocyclooctane, which is an octane compound, which has an alkyne, conjugated with a particular fluorescence molecule. So it gives, it absorbs and emits at a particular fluorescence. If you are able to catch that fluorescence, you can catch that molecule. Using that, if you incubate these cells with this particular octane, after you've added the azide, you start seeing fluorescence on the surface of the cells. So you can see how clearly these cells are, the edge of these cells are lighting up with green. That is because this particular fluorophore molecule fluoresces in the green range. Here, of course, they added a compound, they merely added just an acetylated mannose. And so obviously the octane will never catch the acetylated mannose. They'll only catch the azidomannose. So that's why the top picture is what gets lighted up. The bottom picture, the green doesn't get lighted up. The blue here is just a mark of the nucleus. They just want to mark it just to show that, look, the cell is there, and that this is the cell, this is the cell center, and so around it, whatever has to happen, has to happen. So, the Bertozzi group kept on making more and more of these octane compounds, which are stable, which are non-toxic to the cells, and they are able to bind to the azide that gets presented on the surface. So this is a dibromo, and then this is a bi-aryl azido octane, which they have cyclooctane, which they were able to get the cells to light up with. So different compounds, different cells. This was a different cell. This was a U2OS cell. This was the details, the biological details are not pertinent here, but they are basically able to therefore mark cells. That's the most important point. They're able to use click chemistry to mark cells because it's an extremely specific reaction. Again, as Professor Viswanathan talked about, this is also called bioorthogonal chemistry, because here what they're doing is they are introducing molecules that you would typically not see in the biological domain. So, this goes to that sharpness is, very ironical aspect that let's not try to mimic biology. So alkynes, azides don't exist in the biological sphere, despite all the million years and billion years of evolution that nature has had to work with. And so we have introduced sort of foreign chemistry, something that is orthogonal, something that doesn't interfere with the natural biochemistry at the same time. So in this sense, this chemistry is orthogonal to the biochemistry. It does not interfere with it, and the biological chemistry also does not interfere with this chemistry. And so this allows you to study this almost in sort of in an agnostic manner without affecting your chemistry, which so many of biological techniques and interrogation methodologies are hampered by. You always have off-target effects, non-specific effects, and so on. And this is highly specific, highly efficient. Can we take this beyond cells into organisms? Yes. So here, for example, the Bertozzi group used another molecule, per-acetylated N-azidogalactose. This particular galactose would otherwise go and bind to these very thick sugar that is present on the surface of, say, intestinal cells and all. The kind of sugars that you get in okra that makes things slimy. So that slime coat that is part of many parts of our body as well. And so those slime coats are made of these galactose molecules. And so they use this per-acetylated N-azidogalactose. They put them within embryos of zebrafish. And then they use the probe with the octine to basically go and try and catch the galactose. Could they do that? Yes, they could. As you can see, here is the azido probe, and here is the azido labeling. Here is the simple galactose, acetylated galactose labeling, and you cannot see any fluorescence here, whereas you can start seeing the embryo's head, the zebrafish embryo's head in this as you go along. Okay, so this is one way of now being able to label in space. Can you use that to label things in time? So yes, you can. So what they did is simple. This is a beautiful, elegant methodology in which what they did was, at a particular developmental stage of the development of the zebrafish, or the embryogenesis of the zebrafish, they added one fluorescent compound. Okay, and then say the green in one case. And after some time, in the next stage, they added the red one. So what you get now is, using this green and red, you can say which parts of the body are sort of have the particular galactose in the first stage, and which parts of the body get the galactose in the second stage, using green and red as two markers of two different stages. So using this, they're able to actually capture stage-specific expression of galactose, very, very interestingly, of galactose in space and in time, using very, very specific chemistry that does not interfere with the biological chemistry present within the zebrafish, and thus hence does not affect the zebrafish in any way. The development is perfectly fine here. Okay, so with this, I conclude to show how click chemistry and bioorthogonal chemistry have become very important processes that are now being used for various ways. And I'll conclude by talking about two discoveries here, or rather, well, the first one is discoveries in the fundamental zone. So in the fundamental zone, what kind of new biology are we learning with this chemistry? So what Bertozzi's group showed in a stunning paper in the beginning of this year, which was published in Cell, was the fact that on the cell surface, I talked about glycoproteins, I talked about glycolipids. Nobody had even thought that ribonucleic acids could also be present on the cell surface. And not just that RNA can also be conjugated with sugars. And again, using click chemistry, click bioorthogonal azide alkyne chemistry, they were able to show that a new biological conjugate, glycoRNA, exists and is present on the cell surface and has sialic acids. Thankfully for them, I think the sialic acids were, because that's what they were using, so they were able to show it using sialic acids, and so the tools worked beautifully, but they were able to elegantly show that it is sialylated. Okay. Now, what are the translational implications as far as click chemistry is concerned, where are people taking them? The people are taking them in various directions. But for example, two things that I could think of. One was where people are using them to bring together cancer cells and immune cells. Immune cells would otherwise not be able to see the cancer cells because of various ways by which cancer cells evade discovery. The way people do it is that they have two antibodies. One antibody would bind to the cancer cell, the other antibody would bind to the immune cell. And now you have click conjugates conjugated to the antibodies. So the moment you have these two compounds coming together, they click, and the cancer cell and the antibody and the immune cell are now close to each other and can do whatever they can. The immune cell has a good chance now of taking the cancer cell. Another way by which people are using click chemistry in a very beautiful way, are trying to test it in a very beautiful way, is that you would again have an antibody which would go and bind to cancer cells. Because it's, I work with cancer. This is a domain I'm slightly more familiar with, so I'm trying to give you examples of how click chemistry comes useful in cancer. So what they do is that people have an antibody which will go and bind to the cancer cell. Once it goes and binds to the cancer cell, this antibody will be conjugated with a click agent. Now, you add a prodrug, a drug which is in its precursor form. You add the prodrug, which does not have any side effects. Typically, many of us would know, those who have had the misfortune of having their close ones diagnosed with cancer would know that chemotherapy is a very debilitating condition. Chemotherapy has a lot of systemic side effects. How can we decrease those side effects? One of the ways people are thinking about is again using click chemistry. So the prodrug, there will be a prodrug which can go to very high levels in our blood, and it is not active. It's not active in any way. This prodrug now can be administered to high levels within the body. The prodrug also has the other end of the click. But the moment this prodrug will now go and come in contact with the antibody, which has the other click adduct, now you will have a click chemistry going on very close to the tumor. And there the prodrug gets activated to become the drug. And there the drug can have its effect on the cancer in a more localized fashion. In this way, we can have the drug getting activated very close to the tumor, because the click chemistry being bioorthogonal does not affect anything else. So it happens in a physiologically friendly manner. So that's where the translational implications are coming through in terms of both being able to get cell-cell reactions, cell-drug reactions, and so on. Click chemistry is being used in such beautiful ways. So with this, I end my talk, and I again summarize by saying that click chemistry is intrinsically a very simple and elegant concept, but has groundbreaking implications in the way it has been utilized and is being adapted by various groups all across the world. And it again comes from a very ingenious way of being able to imagine a new world which is different from the world by which, sort of say, nature has tried to use chemistry. So in that way, it is a very brave new world that has been created by Sharpless, Meltal, and Bertozzi, and for that they need to be lauded and richly deserve the kind of recognition that they have gotten this year through the Nobel Prize. So I'll end here, and of course, open up for discussions and questions and so on.
First, we will restrict our questions to the topic on hand, and general questions like, what is the scope of chemical engineering, which even I wouldn't know. But then, any questions here, please? Yes, please. You can introduce yourself and ask the question. Introduce yourself, your name, which college?
Okay.
Yeah, if you come here, you will be on the video camera. Yeah.
Hello.
Hello, sir. Sir, I am Abhinaya and I am from CEG Anna University. I am doing my PG first year in ME Remote Sensing and Geomatics. So, as you said, I would come to the last part. You said that it will be very useful for detecting the cancer cells. Yeah, no? Uh, but my question is whether it will stop the spreading of cancer. That means any stage of cancer.
Oh, yes, of course. This is a very deep question, and I don't know if people have made strategies that will stop the spread of cancer. I think the click chemistry is a brilliant technique to be able to detect it. So, see, the problem is that cancer problem is the problem is once it spreads. Oh, wonderful, wonderful. Sorry, I know you cannot see me. But if you can hear me, that's great. Yes. So the problem with cancer, of course, is that once it spreads, it becomes very difficult to sort of manage it. So I think where click chemistry will certainly come in great use would be to be able to catch it. And again, a big problem of cancer is catching it while it's early, purely because the number of cells which are cancerous at those early cells are so small, so tiny, that the number of chemical reactions that are required to catch it need to be high. And that's where, again, click chemistry would be very useful, because what click chemistry does is that the signal to noise ratio of these chemical reactions is very high, given that they don't occur in the body. So, great. So let that be light. So with this, we would be able to catch those very small, what are called micrometastasis, or the small population of cancer cells can be caught if we are able to engineer them properly using click chemistry. Managing is a difficult problem by itself. So, we are far away from it.
Okay,
Okay.
sir. Okay, sir. Thank you, sir.
Thank you. Thank you so much.
He has covered the something else. No, so in cancer, so what we call it as the metastasis. So is it possible to detect the stage where it reaches the metastasis?
Sorry, is it possible to detect a stage?
State of metastasis. So if you have a cancer in a particular organ, after some time it bursts out and it spread across the multiple organs. What they call it
Yes.
as the metastasis, right?
Yes. Yes.
So can we detect that stage in using click chemistry?
Using click chemistry, yes, certainly. So that's again the advantage. If using if we are able to know some marker, the whole point comes down to knowing a marker that we know is definitely present within cancer cells. The problem again with metastasis is sometimes the cancer cell that starts from, say, organ A and goes to organ B, it may change its behavior, which means it may also change its surface. So if it changes its surface slightly, then it becomes very difficult even with click chemistry to catch it, because you are thinking that it will be putting this particular molecule on its surface, whereas the cell has changed completely. So that is a challenge one has to think about. But if not, if the cell has not changed its properties when it has reached the organ B, then catching it becomes a very click chemistry can be used in a very elegant manner to catch it, because, like I said, the signal to noise ratio is so high that it's literally like what people nowadays use is called the positron emission tomography, PET, PET scan, to using metabolism of cells to catch it. But where click chemistry can go is that even if there are cells that have spread but are not metabolically active, click chemistry can be used to catch those cells for detecting metastasis. Yes.
Thank you. Any other questions? Yes. I have a couple of questions, but I'm going to reserve it at the end.
Sir, I am Gopalakrishnan from Ramakrishna Mission Vivekananda College, sir. Sir, it was almost a splendid lecture from yourself, sir. Sir, I have one very basic question, that in aqueous medium, how come these 1, 2, 3 triazoles that are formed, how come these would be stable, sir? Do we expect the in vivo stability same as in vitro stability? All these click chemistry and other things work out good only when the compound end result would be stable, no, sir? Are we 100% sure that all these click chemistry will work that good even in vivo systems, sir?
Very good question. Yes, absolutely. In fact, I did not get into the details, but there are, and that's where the talks differ from the papers. And since you are asking such great questions, you should not, I would suggest you start reading the papers, because that's where all the some of the hardships associated with these reactions are dealt with in more. So, for example, when the in vivo, when especially the Bertozzi group tried to work these things in vivo, you would see that they have not just used one alkyne; they've used multiple different species of alkynes, and then finally sort of homed in on certain alkynes, because all the alkynes will not give them the same efficiency, the same stability of the cycloadduct, and so on. So eventually, it goes on to keeping on tweaking. Once you have it working, you tweak and tweak and tweak, and that's where the brilliance of an organic chemist comes in here. And until you get the right kind of cyclooctane that you can use to get the perfect reaction without it leading to any kind of unstable processes. So yes, it is definitely possible, and that is what has been demonstrated in now hundreds of papers. Is it efficient? Is it equivalently efficient? Is it equivalently stable? Is it equivalently specific? There is where a lot of effort has gone in, and multiple such sort of sister reactions have been made to show that one can keep on improving these processes and keep on improving the kinetics of such processes. All right.
So we have a couple of questions in YouTube.
Yeah. Or just give me. Ram, there are a couple of questions on Zoom, the Zoom
Sure.
people. I'll just read the question for you.
Sure.
How can we use click chemistry to detect dementias at early stages? Click chemistry
Dementia.
and dementia.
Okay. Yes. These are, of course, very difficult questions, and I'll tell you what the challenge is. The challenge is that more often than not, dementia and dementia-associated diseases are now considered as a part of a larger sort of diseases of tauopathies, if I'm not mistaken. I'm not an expert on it, so you should not quote me on that. But it forms basically due to a bunch of reactions of products which accumulate. These products accumulate over time within cells. As long as they form within cells, plaques and so on form within cells, it's not easy to catch them using click chemistry, because click chemistry, as you'd see, at least the way Bertozzi has used it, it works on cell surfaces. However, if these plaques do come out, and I know for a fact that in some diseases they do come out and form these bigger fibrils and extracellular plaques and so on, then one has a better chance of being able to pick them up if one has antibodies. Again, antibodies that are linked with, say, the azide, and then you put in an octane probe, then one can actually use these probes to detect where these plaques are, even to small concentrations within the brain. And that can help in being able to detect tauopathies and so on. So microplaques and all could be caught. But I would say outside the cells. Inside the cells is challenging right now, is what my belief would be. Again, I'm not a neuroscientist, so take this with a pinch of salt.
Yeah, there's a follow-up question, but I think you have already probably answered that. The follow-up question was, the person says that different types of dementia, and can we classify them, and can we differentiate the different types?
Right. I think a cognitive neuroscientist or a neurochemist would be able to handle this slightly better. Yeah. I am a doctor by training, but it's been a very long time since I read my textbooks on dementia and so on. So I'm sorry about that.
Yes.
Good evening, sir. Thank you for your talk, sir.
Thank you so much.
By using this click chemistry, whether it will be useful for identifying, detecting, and capturing the newly formed viruses, such as COVID came earlier, no, sir? Like that, by using this click chemistry, we can detect them, and shall we do anything for it, sir?
Yeah, I don't know. I really haven't followed up on that literature, but I know that, for example, the Bertozzi group was at some point of time interested in trying to look into this. So, my suggestion is please, help me in this process. I really haven't followed it. You might want to go online and look up whether the octine azide has been used to detect viruses and viral proteins. The problem again is that viral proteins would be present mostly inside the cells, and unless it gets extruded out, catching it might be tricky. So I think that it still needs to be explored, and I'm not aware of literature, but there might be literature. You might want to look this up.
Thank you, sir.
Thank
Next
you
question
so
I have
much.
is, by using the click chemistry, whether we could identify the optical characteristics or particle characteristics of the molecules, sir?
I don't think so, because I think the sizes of these adducts are large. So in a way, one can probably use them to detect tumbling dynamics. One might be able to do sort of fluorescence spectroscopy and so on. So perhaps those things can be done, but I wouldn't think one would use click. There are so many better ways of being able to do it. So the beauty of click is to detect it within more complex biological milieus, and there are but better ways available, like fret chemistry and so on.
Okay, sir. Thank you, sir.
Thank you so much.
Click chemistry can use in preparation of vaccines. Yeah, I think it's a question again with regard to vaccines for COVID. Can it be created by click chemistry? But I think it's the answer to the question that you asked, that you addressed earlier.
Yes.
Okay. If there's nobody, I have a couple of questions, and it's very much similar on the lines to which the gentleman asked earlier, but then I'm looking at it from the side of the alkyne, the very strained alkyne. The very strained alkyne, I think, is what reduces the activation energy, and that's where your thermodynamic driving force really comes.
Yes.
But then the strained alkyne is not just a dipole or a phyl, it is also a dienophile. It can also conduct reactions of, addition reactions across the double bond. So how will you ensure that it goes and looks only as the dipole or a phyl? And even though you said that Caroline has tried many different compounds, but I would still think that whatever alkyne you choose, it's always going to be subject to this dichotomy or polychotomy of these reactions. So how is that really going to work eventually?
No, you're absolutely right. So this is there is a possibility. I think what has happened is, in these cases there are fortuitous discoveries where certain combinations have worked so well. It's also difficult to predict how and why they may have worked when others failed. So I completely buy what you say here, and I think they are still developing chemistries to sort of weed out possible isomerizations that might be taking place in the products and so on. I'm not so sure of the chemical side of it, but you are absolutely right on that.
I think the smart thing was they used the alkyne, and alkene might not have been so strained, and therefore would not have really done the click chemistry as well.
Absolutely. So in the beginning, they were using these these rubber polymer enes like norbornenes and so on. They quickly moved on to alkynes, and then it works.
Yeah, you
wonders.
needed that strain to decrease your activation energy.
Totally.
Yeah, and one other question.
Yes.
I've come across people, I've discussed with people who work on unnatural amino acids.
Yes.
Taking biology beyond the realm of the 20 or so natural amino acids.
Yes.
Would click chemistry play a role into incorporating these unnatural amino acids into proteins and coming up with new biocatalysts, new enzymes?
Yes. So there has been some very good work done in that zone. I have not really explored it. In fact, Bertozzi group itself has done some very good work there.
Okay.
So one can use, as you rightly put it, not just unnatural amino acids, but even non-amino acid conjugations can perhaps also be brought together by click chemistry. That's an entirely new world of chemistry that they are exploring.
Okay.
I have not followed that literature, but it's there. You are right. You're absolutely right on that.
Because the
That
follow-up question I then have is, if you bring in unnatural amino acids, then do you have to repurpose some of the genetic codes to work for these amino acids?
Okay, that will require synthetic biology to get combined with click chemistry. Maybe something for the next Nobel laureate.
Hello. I have two generic questions.
Please.
Is there a way, can I understand it at the theoretical fundamental level, what is click chemistry is doing? At the level of,
Sorry?
at the
Tell me
fundamental
again.
statistical mechanics level, can I understand how the click chemistry is working? Is it possible?
Okay. Yes. So from a very fundamental point of view, what you're getting is are these coincidentally extremely well thermodynamically driven reactions where you have a very strong sort of lock and key mechanism operating. The interesting aspect is that these are reactions which involve chemicals that are known to sort of conjugate with each other very strongly and so form very stable end products. And so these are basically driven in one direction and are unlikely to give rise to other products. So that's at the very fundamental level, that's what's going on. Is it something that they have newly discovered? No. In fact, these reactions have been known for a very, very long time, since in fact, 2 centuries, not 2 centuries, but even before the 20th century, some of these reactions were known. There's another reaction, the Staudinger reaction, which I did not mention, which the Bertozzi group was looking, which is not exactly even a click reaction. And even that involved phosphines conjugating with alkynes, which they with azides, which they very soon realized the click is better than that. So, some of these reactions have been discovered ages ago. But the aspect which is important is that choosing these reactions from among other reactions and also fitting them within the biological milieu has been where the brilliance comes through.
Okay. Very naive question, more like science fiction. Can this play a role in some understanding of speeding up evolution?
Okay, yes, I think, partly the kind of thing that Professor Viswanathan was talking about. So if we can in some way, incorporate these reactions on a more, into biology, then we can. But then remember, the motivation was quite opposite. The motivation was that these are reactions which should not be taking place in any way, anyhow in biology, within biological cells. So they have been chosen to be as far as less fictional as possible. Having said that, it's in the realm of achievable, once we have thought about it. So yeah.
But Ram, can you see, when you look at these cycloaddition reactions, the 4+2s and all those,
Yes,
driven
yes,
by
yes.
Woodward-Hoffmann rules, in some sense that's a click chemistry, right? Because the orbital symmetry clicks these molecules into place and you essentially get stereo-specific molecules. So,
Yes.
So the word click chemistry is new, but the Woodward-Hoffmann rules have been known for quite some time.
Oh, absolutely,
So
absolutely.
I would think that's also click chemistry.
Oh, absolutely. It's also that one of the major things was again the idea that you by using copper they brought the temperature down. That was one very big achievement.
Okay.
Many of these reactions were known. Many of these reactions were happening, but they were happening under fairly exotic conditions and unnatural conditions. So that was one of the biggest things by Sharpless and Melton.
Good evening, sir. I am Priyanka, first MSc Biochemistry in University of Madras. Sir, I have one question. In cancer, you said, sir, the clicks the ducts are going to bind, sir. Is it any possibility to produce the ducts itself as a clicks?
Sorry, I didn't hear the last part. Could you repeat?
Sir, the ducts itself as clicks.
I'm not understanding. I'm really sorry. Can you repeat it slowly?
Sir, the ducts we are using now, sir. That itself we can produce in bioorthogonal chemistry to produce those ducts in click forms.
Dead cells, is it?
No, drugs, sir.
Drugs.
Yeah, yes.
Drugs. Yes. So yes, like I mentioned, the drugs typically are used in a way. See, you can use drugs to come and conjugate with cancer cells and so on. That can be done. But the problem is that these drugs, if you put them at very high levels within the body, they will always have the side effects. So that's why what some people are trying to do, they're not yet they're not succeeded yet. It's not in the market, and it won't be in the market for a long time, I think. Is that they're putting pro drugs as conjugated with these, say, with the alkynes, which they can then sort of bring it close to the cancer cells. And in near the cancer cells, there are a lot of enzymes which can convert the drugs, pro drugs, into more active forms. So that is the way they are using it in such situations. Okay.
Okay, sir. Thank you, sir.
So that camera.
Sir, thank you very much for a very enchanting lecture. Sir, will you please kindly explain what is the chemistry of the brain?
Sorry, what is that?
Chemistry of the brain.
Chemistry of the brain. Sir, that would require me to probably first study and then write a book because the chemistry of a brain cannot be explained in a paragraph. Sir, there are lots of chemicals, thousands of chemicals that are present in the brain. And that depends on the kind of cells that are present in the brain. So again, there are, very different cell types. And these cells communicate with each other through chemicals which are able to transmit information from one cell to another. So that's the way the brain works. In many unique ways, it works faster than other organs of the body because you also have electrical communication, which doesn't, affect, which is not part of other parts of the body. So these are the ways, there are some unique ways in which the brain works. This much I can tell you. And these chemicals are involved in sort of polarization and hyperpolarization of the cells, which transmits the nerve impulses and so on. Beyond that, of course, it requires a textbook to really go through. I'm so sorry. I mean, it's a very fascinating topic, but one that I have not certainly taken up for study at this point of time.
See, the altering the energy is always the basis of chemistry. In any chemical reaction, we are trying to play with the energies. And that's therefore... but in fact, Ram, I have this very, doomsday thing that it would be impossible for us to understand the brain because we need an intelligence greater than our brain to understand our brain itself.
True.
And therefore, we will never understand our brain. That's the
True. So yes, to a great extent, as you rightly say, we can understand the chemistry.
Yeah.
We can understand the biology to some extent. We can understand what proteins are there. So it's like we can understand how the sambar, what are the ingredients of the sambar, but why it tastes so well, that we will not be able to understand.
Sir, again, I have a question. If A and B are two unstable molecules, with the help of this clicks, whether it will be possible for making them stable, those unstable molecules, if possible, what types of molecules and what types of structures could it be get binded, sir?
Okay. This is so click chemistry is. It typically does not work with unstable molecules. I've not seen them, but this is something, again, I think Professor Viswanathan will be able to answer this better. I think typically it works with stable molecules which will now come together to form an even more stable adduct. So unstable molecules, to bring them together using click might be an issue unto itself.
Whether this click engineering would be useful for the self-assembly of the nanoparticles, sir? Is it possible related?
Self-assembly of nanoparticles. Interesting. I have to think about this. I have not thought about it, and it's a very interesting question. You must actually search this. I'm sure. See, the idea is click chemistry is just a chemistry. It's a very simple set of reactions. Everybody with their own level of expertise and their own interest can use it in their field. Ultimately, it depends on how you forge that knowledge of your field with that particular reaction. So, it will be very difficult for me because I'm not in the nanoparticle biology or nanoparticle domain to answer this question. Why don't you just search nanoparticles and click chemistry? I'm sure somebody or the other is intelligent enough to have used it in that process.
Thank you very much, sir. Thank you.
Thank you. Thank you so much.
Same one, huh?
No, I have one question with in Zoom and one more question in YouTube. In Zoom question, the why virus are formed and which method we can use to detect it using click chemistry. That's what I understand from the question.
Again, I really don't want to stick my neck out and talk about virology. I'm not a virologist, and like I said, it would be difficult to. I mean, I don't know how one can use click chemistry to detect viruses, except if one has probably some viral proteins that are being present on expressed on the surface of cells. I don't know if that happens, but if that happens, then that would be the context in which I would think hypothetically click chemistry could be used.
There's one more question which talked about, can we use click chemistry to detoxify methyl glyoxal?
Okay, that's a superb question. Okay, if we can use click chemistry to detoxify methyl glycol, I think I will have to look into the biosynthetic pathway and see how one can again hijack. See, that was where the Bertozzi group again was very successful. They could hijack certain chemical pathways in a very beautiful way by sneaking in the azido groups. So we'll have to think if we can do the same thing with, say, glycolysis. Can we dope it in which case then if the methyl glycol has the click, then we can probably target it using alkynes and so on. So that would be one way. But again, the click has so far worked beautifully in being able to detect things. In terms of detoxifying, one needs a little bit more of a different approach, where one brings these molecules towards either cells that can take care of it, or take them towards liver and so on. So those require slightly more cumbersome methods.
Hi sir, I have a question about the azide group. My name is Mohammed Ashfaq. I'm a PG student of chemistry. I have a doubt about the azide. It is highly reactive and it can undergo like rearrangements and all. So how come it will be in the biological system? Like it can be easily protonated, it can be easily electrophiles can be attacked. How come it is possible to use in the biological system?
Yeah. You are absolutely right. In fact, given that azide is being used here, that was I think at some point of time there was a dip in a decrease in interest in working with azide molecules, because azides are explosives, and they are pretty toxic. So in that sense, there was a lot of worry. Again, using by putting all kinds of very interesting covalent conjugations with the azide, one is able to sort of prevent it from going and affecting the electron transport chain where it is typically known to be. Again, azide is if I'm not mistaken, comes and interferes in the electron transport chain and is a poison there. But here we are talking about making it so big that it doesn't even enter cells, and making it in a synthesizing it in a manner in which it doesn't enter cells, in which case it is able to exert its actions on the cell surface, which is why it will not typically enter the mitochondria and carry out its effects on the electron transport chain.
Any further questions? Okay, if there are no further questions, I would like to thank for his wonderful lecture and the very active discussion session. I'm always nervous after a talk, because if I don't get any questions, I think the talk has really bombed. But in your case, I think the talk was so exciting that it has elicited so many questions. And I think it's a tribute to Professor Ram for that wonderful talk. Let's all join me in giving up.
Thank you so much. It's a privilege to be invited here and to be able to talk.
It's a wonderful privilege. Thanks a lot, Ram. And I also appreciate the audience for this wonderful discussion that you brought in to make this session.
Thank you so much.
I'll now hand it over to Professor Vijay.
Yeah, it's a time for a vote of thanks. Before that, I want to make a couple of announcements. So one is now, we have provided the feedback form both in the website as well as in the physical form available for entrance. Please fill up your feedback in the feedback form. That will help us to improve further. So that's the first announcement. Second one, I would always request you to become a member of Tamil Nadu Science Forum. The annual subscription is 30 rupees per year. So you will get a newsletter from us, our activities, all those things. Please become a member of Tamil Nadu Science Forum. You can do that in the desk, help desk available outside. That's with that two announcement. I also have a link for Telegram. So we have a Telegram group. So if you are joining the Telegram group, you will get all the information about our activities and the programs. Please become the member of Telegram groups. And if you leave your mobile number, other thing, we can include you in the WhatsApp group as well.
Feedback form.
Yeah, if you provide your information in the feedback form, we will include you in the WhatsApp group, as well as we have a Google group for which is around for 4,000 members. So we'll add you in the Google group as well. So we are not going to dump mails. No, it is only a communication. We are strictly moderating the functioning of Google group. Don't worry about that. You are going to get a lot of spam messages. That's not going to happen. So with that, let me thank Professor Ramray. So he has immediately accepted when we sent my invitation to him, and he has accepted it, and he has given a wonderful lecture. Thank you very much, sir. It was enjoyed by the audience, really. And the audience also know, this is the first experience we have a good number of audience for chemistry. We have conducted 4 to 6 lectures on chemistry. Usually the turnout for chemistry is less. This time I see that it is 50 people watching in the Zoom, and another 25 in YouTube. And again, one more live telecast is going on from Anna Centenary Library. And we have audience of around 70 audience sitting here. So it's a good turnout for us, and it is encouraging us to go for more such lectures in future. Yes, sir.
No, I just use one YouTube language for all these people. If you like this, please go share, subscribe, and click the bell icon.
Yeah, that's correct. Yeah, we want to have more such chemistry lecture in future. So if you leave your email ID and other things, no, I see we have a set of audience who are interested in chemistry. Naturally, Professor KSV is going to help us to bring more interesting lectures on that. We will have many future chemistry lectures. So that that is my another announcement. So my next thanks goes to Dr. KSV. He has done a wonderful introduction, and he made the audience very enthusiastic to participate in it. Yeah, that's another interesting thing. And my thanks go to Dr. TRG. He is always a patron for us, and he did all the arrangement for all the PSLs. And he got a special invitation, effort to bring Dr. KSP here. So thank you very much, sir. So I thank all the audience because they patiently sit here and actively participated and ask your question. Thank you very much. And my thanks goes to the Anna Centennial Library. They always encourage us to have such lectures. They keep on providing us lectures, this lecture hall, and we have many such lectures in the past, and we will have future as well. My thanks goes to Anna Centennial Library. With that, let us close the meeting. Thank you very much.